Hi, my name is Tammy, and today I'm going to be talking about DNA replication. So in our first step with our basic knowledge of DNA, it's formed in the double helix structure, as you can see right here, where it has two lines and it turns. And it's made by, there's a phosphate sugar backbone right here, and there's carbon, and there's a carbon ring with 5 carbons in total. And right here we have two complementary base pairs, and the base pairs of complementary ones are adenine, thymine, and cytosol and guanine. So it's ATCG. And the phosphates and the carbon, it's formed together with the phosphodiester bond. And between the base pairs, there's a hydrogen bond, which is actually pretty weak. And this entire thing that you see right here is a nucleotide. Okay. And for the next one, so as the process starts, there is an action helicase which actually unzips the two strands, and it breaks the hydrogen bonds in between the AT and the CG, as you can see right here. So this right here is helicase. And for our next step, let me just move this over here. Okay. So there's RNA primase, which produces RNA primer. And as you can see, this right here is still helicase, and this is helicase. This is the primase, and this is the primer. The primer basically serves as an anchor so that the two strands do not close while they're trying to replicate it. Okay. In our next scene, we have DNA polymerase 3, which assembles a nucleotide. So as you can see, the purple one and the ring right here, these are both the DNA polymerase 3. They go through the strand, and this is the leading strand right here, so from the 5 to 3 direction, it assembles nucleotides together. And in our last scene, so as I mentioned before, this right here is the leading strand, so it's from 5 to 3, and DNA polymerase 3, which assembles all the nucleotides, and on this side, this is the lagging strand, which is from 3 to 5. And so as you can see, there's actually a RNA primer for every single Okazaki fragment that I put in there, because every single nucleotide has to be connected to the third carbon. So as you can see on this side, this is the lagging strand, and the DNA polymerase is putting on the DNA fragments slowly, one by one. And the helicase is still right here. So after it's completed, the DNA polymerase 1 removes the RNA primer, especially from the lagging strand, because for every single Okazaki fragment that's been implanted, an RNA primer has to be there. And for the final step, a DNA ligase is used to glue all the different Okazaki fragments together. As you can see right here, this blue dot is the DNA ligase. And right here we see that, so this was the leading strand, and this is the lagging strand, and it's a semi-conservative replication because half of it is new and half of this is new. So that's it. That's the process of DNA replication. Thank you.